we have got Chloe Boote, who is a coral scientist and a PhD graduate from James Cook University in Australia. She's originally from the UK and volunteered in aquariums and teaching reef ecology in Honduras before her PhD. So Chloe is going to be joining us, I believe, from Australia today, and we'll bring Chloe in to confirm. Are you in Australia?
Yes, I am in Australia. I was going to say good morning from Australia, although I think it's your nighttime now. But yes.
Yes, it's nighttime for me, but it's tomorrow where you are.
Yeah,
So
well, thanks for having me so late.
we're very excited to have you today and hear more about all of your work with coral reefs. So with that, I will bring in your screen, which looks good to go, and I'll let you take it away.
Okay, well, thank you. And firstly, I just want to say a big thank you for having me. My name's Chloe, and currently I work at the University of Technology, Sydney. And I work with Emma Camp, who was meant to present today, but unfortunately got COVID. So I'm a last minute fill-in, so I hope this is okay for you all. But today I'll be talking to you about what I did on my PhD, which was with James Cook University and the Australian Research Council Centre of Excellence for Coral Reef Studies. Now, firstly, a little about me. I did my undergrad in the UK at Portsmouth, and I managed to get funding to go do an honours project out in Indonesia, where I was looking at the heat stress of Porites lutea, a particular coral species, which was a really exciting experience because I had to learn to basically build all my own equipment because they don't have a lot out on a remote island in Indonesia. And this went really well. And following this, I went to do my master's at University of Essex. Again, I did a little trip to Indonesia, but I also did a project on looking at heat and light stress response in a coral called Aquapora digitafera. And this is where I started getting into more genetics because I'm really obsessed with genes and microbiology, and I think it's all really cool. And then again, following my master's, I went out to Honduras, where I worked as a reef ecology lecturer, teaching students how to do reef science. And also then I went to both the London Zoo and Horniman Museum and Gardens in London, and I volunteered as an aquarist, so working in an aquarium, and also doing outreach and talking to people about conservation. Now, following this, I was very lucky to get a scholarship to James Cook University. So I packed my bags and I moved to the other side of the world, just like that. Just thought it'd be a good decision. And I went out and I got to do all my field work in Orpheus Island. So this here is Orpheus Island, absolute beautiful spot of the world with amazing reefs. And I got to study these, and they've got fantastic facilities. So I could do a PhD looking at coral spawning and developmental biology. And then also a big thing of mine, like looking into coral bleaching, and again, that genetics and microbiology. So I completed this in February, and currently I'm working as a research assistant at the University of Technology, Sydney. And here I get to do cool things like reef carbonate budget surveys, where I get to survey the reef and see really cool fish. And I also get to do some stuff with the Coral Nurture Program, which I believe Emma spoke to you guys about last year, outplanting coral fragments on the reef, trying to regrow the reef, with lots of amazing people for different tourism operators out there. And I also get to do some really cool stuff in the lab. So that's a little about my background and my journey. And then now I suppose I'm just going to talk to you about my PhD research, get into the nitty gritty of the science I do. So firstly, reefs, coral reefs, they're super important, beautiful ecosystems and so biodiverse. They support thousands and thousands of marine species. They support us with coastal protection, food, livelihood, and they also do an amazing amount for nutrient cycling in the environment, which is really important, particularly in today's climate. And they also have some cool products on them for biotechnology, so looking into medicine and future products and all this nifty kind of stuff. And they also, though, unfortunately, are threatened quite severely for numerous different threats. So we have local threats like over-exploitation, oh God, I can't say the word, so like overfishing and pollution runoff and invasive species. And then we also have climate issues like ocean deoxygenation, ocean acidification, and also ocean warming, which has always been a big thing that I've been concerned and want to learn more about. So this beautiful reef here in Indonesia, and then unfortunately we're seeing this rising ocean temperature, which is leading to large-scale bleaching across the globe. So here is a picture that Greg Torder took during one of the JCU bleaching surveys in 2017. This is me just swimming over the reefs at Orpheus that bleached then. And it's really sad to see when you're like, love this ecosystem, that you get these beautiful, colorful reefs, and then suddenly all the color's gone and they're not very healthy. And it's funny, this image has been snapped up by a lot of media people. I see it on articles all the time. I'm like, oh, it's me. But yeah, so we get these reefs and they bleach when they get too hot. And unfortunately, if they can't recover from the bleaching, we actually see that the corals will die off and we lose this reef. We lose all this biodiversity, all these big, beautiful structures. They're gone and they'll degrade down. And we no longer have all these amazing, ecosystem functions that we get from reefs. So my thing was like, oh, why is this happening? What is that genetic mechanism, or what microbes are involved in this bleaching response? And there's lots and lots of theories about it. But one of the things that I was really interested in is the fact that corals have 2 tissue layers. So us as humans and other mammals and we have 3 tissue layers, but corals, because they're less evolved, they have their outer skin layer, their ectoderm, so that's their outside layer. And then they have their internal layer, which is their endoderm. And this is separated by a sort of liquid barrier, the mesoglia. But the really important thing is that these Symbiodiniaceae or zooxanthellae or the photosymbionts, they live within this endoderm. So they live in the inner tissue layer and they live in symbiosis with the coral, and together they are really important for coral health functioning. And the coral pass products to the algae, the Symbiodiniaceae, and the Symbiodiniaceae pass stuff back to the coral. So what we have when the corals experience heat, unfortunately, they get really stressed and they start to produce toxins that are really bad to be in their cells. And then, so from this, the Symbiodiniaceae are then either expelled out of the coral or they are engulfed. And we don't particularly know exactly if it's the host that kicks this off, the coral, or if it's the algae. And we really wanted to look into this endoderm region because it contains the Symbiodiniaceae. So we're like, well, this must be the point where bleaching might happen. But it's really hard to study in typical coral models that we have. So the photo I took of Acropora millipora, again on Orpheus Island. Now this is the thing, so colonial corals, or most normal corals that you see on the reef, they're made up of thousands of tiny, tiny little individual corals living together as one big colony. So kind of like a beehive, like there's all these tiny, tiny little polyps, corals living together, and they're working together, and that's how they are. So it's really hard to look at the endoderm when it's so, so, so small. So we had this idea of creating a new novel coral model, and this is what we proposed. This is Heliophangia Actiniformis. Now this here, it looks like a sea anemone. It looks like a giant sea anemone, but it's not. It's actually a coral. So this is where this unique thing comes from. It's good to be unique. It's good to be weird. This coral is just, it's got these big tentacles. It's large. And the really interesting thing is that because it is so big, this is here an adult, about the size of a dinner plate. It's bigger than my head. And it's one large solitary coral. So as I said before, with the polyps that are all interconnected, they're all living together, they're really small. This is actually just one huge coral polyp. And the other thing that's cool is it's not attached to the reef. So most corals are attached, they're stuck on the reef, but mushroom corals, which Heliophangia belong to, can actually move around the reef really, really slowly. And then Heliophangia is very unique within mushroom corals because it's got these big tentacles, but it also has one really big mouth that is so large it can actually eat fish. So this is a photo taken, I think, from National Geographic in 1973. This is a tiny little damselfish having a really bad day because it's about to be eaten by a Heliophangia. So yeah, that's all what my PhD was about. For the first time in coral biology, We could physically separate this ectoderm and this endoderm, which was methodology taken from one of my supervisors, Aurelie Moyer, that she did on anemones, and we applied it to Heliophangia. Also, whilst working on it, I realized I could look at different regions. So I could look at the mucus, which is this outside layer on the coral, like this secreted slime all over the coral. I could swab it and have a look at the different microbes going in on there. And then also I could look at that ectoderm, the outer skin layer, and the mesoglea is that liquid in between. And then we have the endoderm with the Symbiodiniaceae living within it. I also realized during my PhD that Heliophangia are unique as they have nematocyst bundles or acrospheres. So this is all the little stinging cells that live in corals and jellyfish. Rather than being throughout the coral, they're actually all isolated to these little tips of the coral. When you go near it, it's like a medusa head, like they all come towards you to like sting you with them. And also I realized that I could actually sample the gut because it's so large. And finally, another thing that comes from using a big coral is that it's really good for microscope work. So this is a microscope image. I think easiest thing just to take from it is that this is one of the tentacles. This is a slice of the tentacle skin. And what we can see is it's really clear. This is the ectoderm, this outer layer. We've got this mesoglea in between, this like liquid barrier. And then the endoderm here, this inner layer, which is packed full of Symbiodiniaceae. So those tiny little algae photosymbionts that live in the coral, they live here in the endoderm. And it's really good because you get such good resolution, because with all the tiny coral, the colonial corals, it's all so tight and smushed together, it's really hard to see. So what did I do? I had a non-destructive method of sampling. I'd collect my corals, and I had multiple sampling points from 2016 to 2018. And I would go collect my corals and I'd snip off a few of the tentacles, which sounds a bit mean, but it's kind of like cutting their hair. Like they're used to losing them in the wild. It's not a big deal for them. And I would take their coral tentacles and I would dissect it into the different tissue regions, and I'd look for microbiome and host genetics. This is me just doing my little dissections here. And then we also have me doing my mouth swabs. It's really funny because, yeah, you could cut the tentacles off the coral and they weren't bothered. They're like, yeah, whatever, do that all the time. Stick a swab in an animal's mouth? No, they did not like that. They like physically looked like they were having a sulk afterwards. And then they would recover, and I'd put them back on the reef and I'd go get them next time. So the microbiome results, just showing you some of my science here. The microbiome is essentially all the microbes that live in a certain area. So like humans, we have our gut microbiome and our skin microbiome, and these are all really important for our health. It's been shown that corals have a microbiome and that they also contain microbes that could be really good for their health or really bad for their health. But again, it's like an overall coral microbiome. We don't know where specifically these microbes live, and therefore it's very hard to know their function. So I wanted to look at where my bacteria lived, and I showed here, does the microbiome differ between the tissue regions? And over here on the left, we have the alpha diversity, which is the number of different ASVs or bacterial potential taxa living in that region. So the mucus here has a much higher diversity in the red group, and the ectoderm has a much tighter, smaller community, and then the endoderm is somewhere in the middle. And then we have the beta diversity here on the right, which is really interesting because it's not just the number of species present, but then the abundance of that species. So you could have 5 of this bacteria and 5 of that bacteria and 5 of this one. And this makes up the community composition. And what we can use these plots for is essentially if the groups don't overlap, we know that there's something different. We know that the community composition is different, and then I can look into what is different and what species are different. And again, we see the ectoderm here, this blue one is very tight-knit, very close community. It's not very diverse. There's just like maybe one dominant bacteria. But then the endoderm in gray, it's a lot more diverse as well, with the mucus in red. All these little points are a different sample. And this was really exciting for my PhD. I was like, this is amazing. I'm massively biased, but I was so excited. But I thought, is this just a fluke? Is this a one-time thing? And it wasn't. This is across my 6 sampling points, and we saw the same pattern. We saw the alpha diversity, this much higher diverse mucus, and really like closed off ectoderm. And again, in the plots, we saw that the ectoderm remained quite niche with the endoderm close by, but with some variation. And the mucus just had a really diverse and really variant community, which is not surprising considering the mucus is outside of the coral and exposed to the seawater. So the main takeaways from that was I saw a significant difference in microbial communities. So the ectoderm was its own community, and then the endoderm and the acrosphere, that nematocyst bundle. I didn't show them in the plots because I only added them in 2017, but they had a very similar microbiome to the endoderm, but this was still very different to the other regions. And then the mucus and gut had very diverse and very different microbiomes to the other regions, but again, are much more diverse because they're these semi-open systems and they have the seawater. Another paper that's coming out of my PhD was when I specifically looked at the bleached microbiome and how the bleached tissue regions differed from the healthy, and they showed a really big difference. And the bleached microbiome is really cool because we saw a lot of microbes that we know are to do with disease and being unhealthy, and the healthy ones showed a lot of microbes that are more related to probiotics and being a healthy animal. So it's really interesting to see, oh, this microbe lives in the endoderm, which is a really important region for bleaching, and it's really helping the coral's health. So maybe if we give this coral more of this microbe, and like a probiotic pill or whatever, like a lot of researchers are working on these days, then this could help their health overall and help them survive bleaching. And also just some cool images. So I did fish, not the cool things that swim in the sea, but actually fluorescent in situ hybridization, which is a way of just like validating my sequencing data. Like, I'm not making this up, guys. Like, these are some pictures. So I looked at, so this is just a couple from the ectoderm, and I use these fluorescent probes to tag bacteria. This is a general bacterial probe, but I also use species-specific probes. And then I could find these bacterial aggregates, these big fluffy clusters in the skin, and be like, oh, this is where it lives, and this matches my data. And again, you get this great resolution because here at 100 times, you can actually see like the little fluffy bits that are the different bacteria living all together in the coral. And again, shown to be like specific to the ectoderm. And then finally, guys, my genetic results, just another picture of Orpheus, so beautiful. So similar idea here. The main question was, when Heliophangia is bleached, does it have a different genetic response to when it is unbleached? So unhealthy, healthy. And again, going back to that tissue region idea, is I really wanted to know, is the endoderm going to have the biggest like change in gene response because it's got the symbiodiniose and then it doesn't have the symbiodiniose? And that's what we showed, that the endoderm did have the greatest gene response. Long story short, I did some maths, hate maths, but I managed it. And we looked at which region, so the ectoderm versus the endoderm, comparing the bleached and unbleached groups, like which genes like had the most and the most extreme response, and then which region did that relate to. And what we wanted to see was here, this endoderm group, the main thing to take from this plot is it is a higher value, showing the greater change in gene expression, and it also has a much wider variation. So like 0 would be the normal, genes don't change expression. What we want to see is lots of genes doing a really extreme change in the endoderm. This was also really useful, this interaction value, because when you get gene expression data, you get thousands of genes. Like, I'm talking 30,000 genes. And I'm like, well, I don't know which ones are important. But using this sort of maths to this equation to work out which were the most important genes when bleached and unbleached in the different tissue regions helped us identify the key genes related to this loss, this process of bleaching, which came back with 156 significantly differentially expressed genes, which is a lot easier to work on than 30,000. And it was really interesting. The main thing to take from this is just that they had different expression patterns. So we could see the genes that were only expressed in the bleached ectoderm or only in the bleached endoderm, or if they were across them in multiple regions and multiple areas, or if they were really expressed when the coral was healthy. And this helped us understand the function of the genes because we knew their region and when they bleached or when they weren't bleached. So for example, one of the really exciting groups here is the genes that were expressed at a really high level in the bleached endoderm, so that region that contains the Symbiodiniaceae. And what we see, this is just an example of some of the genes, but like I said, like they're just really highly expressed in the bleached endoderm. And these genes are related to things like immune response. So there's a lot of research on immune response and the loss of Symbiodiniaceae, because these guys might remove the symbionts during bleaching. And we saw anti-apoptosis. So apoptosis is quite a sad process that when corals and cells in an animal are too stressed and they can't handle life, they basically give up and they die. And there we saw genes in the endoderm that were really anti-apoptosis. So they were trying to stop the cells from dying, which is massively important because these are the kind of genes that can help us know what, corals that bleach and die, they don't have enough of this gene. And this might be something that can help corals in the future. We also saw the genes that removed harmful products or toxins. So again, that heat stress on that coral, the coral's really stressed. It's like feeling quite toxic. And these genes are the ones that like, okay, let's take this poison like out of your body. Let's get rid of this. Let's make you healthy, trying to survive because Heliophangia is good at surviving bleaching. And finally, the return of homeostasis. So what genes we need to see to get the coral back to functioning, normal functioning, getting its color back, feeling healthy, feeling good and living and continuing to be this biodiverse ecosystem. So yeah, overall, guys, my tissue-specific analysis can give us a better understanding of coral biology in normal and stress situations. And also, it's really important thing that we can localize this, genetic or microbial response, seeing what is happening in what area when it's bleached or healthy. And then just for fun, as I didn't talk about developmental biology, but here is a really cool video of my corals spawning in 2017. So this is a female coral. These are all eggs. My corals have split sexes. And this is about 1 a.m. and all of us very excited to see this happening because it's such a large scale, because it's such a huge mouth and it basically just vomited out all its babies. So yeah, thank you for listening, everyone. I hope you enjoyed that. And yeah, any questions?
Great. Thank you so much, Chloe. That was really fascinating. A lot of really detailed work that you have done on the corals with your PhD. I'm wondering, kind of with all of the work that you've done and the data you've collected, what do we do now? So we've kind of seen kind of their difference in the bleach and the non-bleached corals, and I understand that like in a lab, you can kind of simulate how you get healthy corals and bring these bleached corals to be healthy again. But how would that translate into the ocean?
Oh, that's a lot. There's a lot of ways. I suppose there's also the genetic way and there's the microbiome way. So there's a lot of great research being done that's looking at, these microbes that are really good for coral health, and essentially like looking at if we can then like put this into the water column, more of these healthy microbes, so that when the coral is stressed, that then they sort of have these microbes to fall back on and they can help them survive. And then also a lot done with like breeding, like baby corals being exposed to, again, the microbes. So when they grow up, they've got it in their microbiome, like we do as babies, we get from our mother's milk or whatever to like survive and be hardier when we're older. The genetic front is more interesting, I suppose, in the fact that there's all these things to do with epigenetics and assisted evolution. So again, either the breeding the genes into the corals or even trying to genetically modify the corals. But there's a lot of ethical stuff that comes with that, because obviously, modifying an animal. But it's all about trying to target what the process is that causes the bleaching, so that then we have an idea of how we can stop it. I don't know if I answered your question or if I just went off on that. But yes,
No,
implementing it into more corals, using this model organism so we know what happens in the colony corals, that obviously we can't look at that tissue region, and then hopefully be able to understand more about it and see it into the ocean.
Yeah, excellent. Do you see kind of the diversity in the mucus and the ectoderm and the endoderm? Is that in those microbiomes? Is that similar in the species that you studied in other species, or are those totally different amongst coral species, or do we not know?
We don't know. This is the first time it's been done with corals. There's some anemones and Nematocella and Hydra that have looked at particular tissue regions, and giant clams as well have shown. So the ectoderm is basically dominated by one species in Heliophangia, and that's seen similarly in other marine inverts like clams and Hydra and anemones. But for the actual tissue regions in comparison to coral, there isn't a comparison. So, I mean, actually, no, there are some comparisons with the coral mucus, because obviously that's the outside stuff so you can study that in corals, and that's really similar to what we see in other corals. But yeah, looking at the guts and the tissue layers themselves hasn't been done before. But it's just very exciting, particularly in the gut, that we had quite a few species that are well known for digestion in fish and reef fish, and so I just got really excited because I was like, oh, it's actually got like a digestive microbiome. But yeah, okay, I'm trying not to be a nerd right now.
No, that's what this is for. This is the perfect platform for
Yeah,
the cool things in the gut of corals. So, I mean, it is fascinating.
Oh, thanks. I think so.
Yeah. So the last we've time for one more question, which is what's next for you? What are you working on currently, or what is next that you're studying?
Next, well, at the moment, yeah, I'm working with Emma, and I'm working on quite a few interesting projects with the elementome, which I think she's going to talk to about today, how the elements impacts the coral, its genetics and its microbiome. And then also, again, I'm looking at other coral species and their genetic response to bleaching, and also her super corals, so ones that work in the mangroves and the reef. At the moment, I'm just trying to finish up and publish my PhD papers so that I can get some postdoc funding and hopefully start a full-time project. So at the moment, I'm just working on a lot of cool, interesting projects. So yeah.
Great. Well, thank you so much, and thank you for sharing all of your work with us today at the Global BioFest. And I hope you have a wonderful day over in Australia.
Thank you. I will. Thank you so much for having me. This has been fantastic.
Absolutely. We'll see you soon.
See ya.